Hello, I'm Dr. Stephanie Cole. As part of my role at Nanotemper, I spend a lot of my time talking to our customers and other researchers in the biologics field, learning about their struggles. As a member of the product squad, I think a lot about how to improve their daily workflows in the labs with new technologies. So I'd like to share with you some of the stuff I've learned over the years, and in particular, talk about one of the biggest struggles I hear about from biologics researchers. Many of you share a similar struggle: how to pick the right candidate so you're not investing time and money on therapies that will ultimately fail in the clinic. There are many approaches for improving candidate selection workflows and making them more efficient, but today I'd like to focus in particular on a biophysical technique called dynamic light scattering, or DLS. So if you're already in the biologic space, you're probably familiar with the concept of developability or candidate profiling. And if you're not, this is just a way of saying that when given a huge library of engineered therapeutic proteins or buffer solutions to screen, you have to figure out which candidates and conditions are most likely to succeed downstream and give your complex drug the greatest chance of making it to market. So there are a pile of techniques out there used to do this work, and anyone in that space will tell you they have a favorite set of criteria they use to choose their best candidates. Many of you out there may already be familiar with DLS and what it does, but I've also heard feedback from many in the field that they don't always know how to add it to their selection workflows in order to get more information about these biologics candidates and their formulations. So today I'd like to show off some of the great information DLS provides to biologic researchers and how it gives insight on the stability of your candidates. With DLS, teams find information about the colloidal stability of their samples. That is, how likely they are to remain well folded and therefore active in solution without aggregating and crashing out. As biologics researchers, you know that aggregation is a big no-no for anything in the clinic, and avoiding candidates and conditions that lead to aggregate therapeutics is crucial for quality. I want to start by giving a really brief introduction to DLS, and then we'll talk about what it tells you and how that information helps you with different aspects of your workflow, with some specific examples mostly around formulation screening. I'll be sharing examples so that you can see what I'm talking about throughout the presentation. Now, some of you here might be new to DLS, and I'd like to start with some of the technical aspects of how the methodology works. I'm not going to get super into the math or physics required to get this data, but I'd like to provide some basics so we're all on the same page. And if you already know how DLS works, don't worry, we'll get to the workflow benefits soon. Dynamic light scattering uses the natural Brownian motion of particles in solution to find information about their size and purity. When light enters a sample, it bounces off the particles in the mixture at all different angles. Now with DLS, the reflected light is measured at a fixed angle for intensity. And specifically, we look at how that intensity changes as particles move into and out of the detector window and bounce that light. Big particles move more slowly compared to smaller ones and stay in the detection region for longer. Therefore, light from large particles fluctuates less than that from small particles. Now, all this information is collected by the instrument you use for the measurement, which then applies a mathematical fit called the autocorrelation fit, or ACF, which deconvolutes that intensity fluctuation information. From that fit curve, software algorithms determine the particle sizes in solution and provide you with the size distribution plot, which you will then use to assess your samples. Now, if you want to learn more in depth about how exactly that information is obtained, please check out the links that we have to other videos called the DLS Easily Explained and the autocorrelation function, the key to understanding your biologic molecules. Those are available in our resource center. They were recorded by our product manager, Leah, and she does a really great job of breaking down where exactly the data derives from and how the ACF is processed to become the parameters you'll need for analysis. Okay, so now speaking of these parameters, you're probably wondering, what does DLS actually tell me? There are two key pieces of information that come out of the data collection: the RH, or hydrodynamic radius, and the PDI, or polydispersity index. Let's discuss the PDI first. Aggregation is a big concern to biologics researchers, as I've already alluded to. Aggregates lead to inactive therapeutics, reducing the efficacy of your treatment. Further, the presence of aggregates is potentially toxic, which is important to avoid when developing biologic treatments. Finally, even the presence of a few aggregated proteins triggers a chain reaction and more aggregation, which makes them less stable and effective over time. The PDI values provided by DLS are great to quickly assess whether you have aggregates ruining your sample. DLS doesn't just tell you about the size of the particles in solution, it also tells you how many different size populations there are and how uniform in size a given population is. It's most desirable to have a monodispersed sample for further development. That is, a sample that contains a single species of protein, well folded and uncontaminated with aggregates. The size distribution peak derived from the ACF fit will be nice and sharp. A monodispersed sample has a low PDI value closer to 0. On the other hand, a poor sample will be polydispersed and have a high PDI value. The exact cutoff for what's considered a high PDI value is going to vary based on what system you're using, but in general, something in the 0.3 to 0.4 range and above is considered very poor sample quality. There are a few ways a sample can exhibit an increase in PDI. On the left, we see an example of peak broadening, which increases that PDI value. Peak broadening occurs when all the protein molecules in that sample aren't uniformly folded, or when there are multiple species present, but they're within a few nanometers of size of each other and can't be resolved as distinct populations by the software. That overall peak height decreases and the width of the peak's baseline spreads out. Now on the right, we have an example of multiple populations present in the sample. Though we see 4 distinct populations, again, the software often cannot distinguish between populations in a single sample within about a half order of magnitude of each other. That is, if you had a sample that was half 6 nanometers and half 8 nanometer radius particles, it's just going to look like a broad peak around 7 nanometers. However, if instead you had half 6 nanometer and half 30 nanometer particles, their intensity distribution graph will probably show separate peaks, although the baselines may overlap. Now, in this example I have here, the purple arrow indicates a very low molecular weight particle, a sugar, a peptide fragment, a nucleotide, some other small molecule that's present in the sample that's about 2 nanometers in radius. The blue arrow indicates the expected protein peak. However, you can already see the peak broadening here, which, as we discussed, is a bad sign for your stability. Finally, we have the black arrows, which are indicating large aggregate clumps. These multiple populations together are increasing the overall sample PDI, and more importantly, they indicate that you have big problems with your prep. A sample with these characteristics needs to be reformulated or thrown out altogether. Now, another thing DLS tells you is the size of your particles. While the hydrodynamic radius may not tell you much for a single sample in a vacuum, when you're comparing all those conditions and constructs, it's helpful to look at. The size of any biologic treatment is an important consideration when it comes to cellular uptake. And that's often a more downstream consideration than what you may be thinking about during early development, but it's still really useful information to collect very early on. Now, in particular, those working in antibody drug conjugate development might be interested in seeing how the conjugation process, which can be quite stressful to the antibody, or the conjugate molecule itself, impacts the final size of the product. So assuming you have a monodispersed sample, DLS will give you a fairly accurate size for the hydrodynamic radius. That hydrodynamic radius is assuming that your particle is a sphere. So if you have a long and narrow protein, the radius will correspond to the longest side. As an illustration, here's an overlay of 2 different monodispersed samples. These samples were collected at separate times and placed on top of each other for clarity. In red, we have a particle that is 60 nanometers in diameter or 30 nanometers in radius. And in blue, the particle is half that size. You can see how the difference in size impacts that decay rate on the autocorrelation function and how that is then reflected in the size distribution graphs. Okay, so now that we understand where the data comes from and what it looks like, and we've seen a few examples of what it's telling you about your samples, let's take a look at 3 real-life examples of where dynamic light scattering is helpful in characterizing candidates and conditions. Monoclonal antibodies are some of the most common biologics researchers are interested in. Here's an example of an antibody we ran in-house. It was left on the bench for a few days before this measurement was taken. Obviously, this is an extreme example, but it shows just how much the environment impacts your protein-based therapeutics. Let's look at the really obvious problems in the data. The expected protein peak is broad and furthermore very low intensity, which means a lot of the sample is not in that population and no longer remains a single tightly folded species. There are other higher intensity peaks at very high hydrodynamic radii. This is an indication of aggregation clumps. Finally, the overall PDI is quite high at 0.97. All of this information together tells you that that's a very bad sample, and it's a good reminder of why we don't leave protein samples out at room temperature. So next up, we see an example of a vaccine antigen protein in 3 different buffers. Vaccine antigens must be optimized for proper folding, so vaccines can be developed to target specifically this disease-triggering protein. The blue buffer is quite obviously bad. There's almost nothing but an aggregation peak. In the case of the buffer in black, a sugar was added to stabilize the protein. You can see the small size peak corresponding to that sugar. Unfortunately, the sugar didn't do enough good because there's also a peak corresponding to aggregation, and therefore it is not an ideal condition. Here we have the buffer corresponding with the sample in red as our best option. You can see that there is a single sharp peak at the expected size for the protein. Now last, I have an example of an accelerated stability study. Biologics teams need to predict how their candidates will behave long-term, and accelerated stability studies help with that. A biologic is stressed for a relatively short time period, and then the information is extrapolated to predict how it will behave long-term. Here we have Herceptin, a monoclonal antibody cancer treatment, incubated in 2 different buffers at high temperature overnight. The DLS size distribution plot clearly shows one buffer is protective, while the other results in peak broadening and increased PDI. The sodium acetate buffer protects the colloidal stability of Herceptin even at high temperatures, which is a good indication it's a better buffer formulation for long-term cold storage. I hope these examples have highlighted just how helpful DLS is when you're making judgments about your candidates and your conditions. DLS is super valuable if you're trying to throw out bad candidates and conditions early on. Anything that isn't stable will aggregate and crash out of solution, and rather than waste time trying to optimize it, sometimes it's less of a headache to choose a different option or start over. In early development, you're also often trying to get through a lot of candidates and conditions quickly. It's worth noting that DLS experiments are pretty speedy. Throughput varies by system, but it's generally possible to screen hundreds of candidates per day. This is great if you want a fast and efficient method for eliminating the least viable candidates. Anything with lots of aggregation or super high PDIs can go straight to the bin, no wasting time on it. Another reason researchers turn to DLS is that it doesn't require a lot of materials to run. Even if your proteins are easy to express and purify in bulk, demands from other teams mean that you don't always have a lot of protein sample to work with. And there's value in having an assessment tool that gives you a lot of information without using a lot of material. So thanks so much for watching this video.